Assalamu alaikum. I am Dr. Sabikun, Associate Professor, Department of Biochemistry. In my previous lecture, I was discussing about simple lipid. Today, I will continue the lecture. Today, I am going to discuss about the complex lipid. There are various kind of complex lipid and there are various important complex lipid. For example, phospholipids, lipoprotein. So, today I am going to discuss about the complex lipid. At first, phospholipid, the composition, the general structure of phospholipid is the fatty acid plus alcohol plus phosphoric acid plus minus nitrogenous base. That means, in phospholipids, nitrogenous base may be present or may not be present. So, the alcohol portion of the phospholipid may be glycerol or sphingol. If the alcohol portion is the sphingol, then that phospholipid is known as sphingophospholipid. Nitrogenous base is either choline or either ethanolamine, or in glycerophospholipid additionally there may be serine may be present. Serine is also a nitrogenous base. Fatty acids are the unsaturated or saturated. For example, in sphingophospholipid saturated fatty acid is usually present. Glycerophospholipid. So, in a structure of glycerophospholipid there is one glycerol, two fatty acid and another carbon atom is attached with phosphate and polar molecule. Examples of glycerophospholipids are lecithin, which chemical name is phosphatidylcholine, kephalin, which chemical name is phosphatidyl-ethanolamine, phosphatidylserine, lipocetal, which is known as phosphatidyl-inositol, plasmalogens, cardiolipid, which is known as diphosphatidylglycerol. Sphingophospholipid. So, in a sphingophospholipid, what is present? Is sphingol, a fatty acid and a phosphoric acid. So, function of phospholipids are they constitute biological membrane. The main component of biological membrane is the phospholipid. As we know, biological membrane is the lipid bilayer, biological membrane composed of lipid bilayer. So, this lipid principally is the phospholipid and provide arachidonic acid to form eicosanoids. Eicosanoids are very important chemical mediators in our body. In bile, solubilize cholesterol and prevent gallstone formation. Helps in coagulation. Dipalmityl lecithin acts as lung surfactant, act as second messenger for hormones such as phosphatidyl-inositol. Plasmalogen act as platelet activating factor or PAF, helps in lipoprotein formation. Glycolipid, the general structure of glycolipid is fatty acid, sphingol plus sugar. Sugar is the hexose sugar or amino sugar or sialic acid and fatty acids are saturated or may be unsaturated. There are two types of glycolipid. These are cerebroside and ganglioside. In cerebroside, hexose sugar is either glucose or galactose and in ganglioside complex sugar along with the hexose sugar present. So, glycolipid, if I talk about the structure of glycolipid, there is the ceramide portion and the galactose. In this case of galactose cerebroside, in case of ganglioside ceramide, there will be a oligosaccharide attached. Importances of glycolipid is these are the structure. Glycolipids are the structural component of brain and myelin sheath. Constitute biological membrane, they also constitute the biological membrane. In biological membrane, the glycolipid always attached with the phospholipid or glycoprotein with a covalent bond, act as cell surface receptor and regulate the cellular interaction and intracellular communication, regulate cellular growth and development, act as blood group substances and tumor antigen. Sulfated glycolipid are known as sulfolipid. And the very important complex lipid is the lipoproteins. These are the actually helps in to transfer the lipid from one organ to another and lipoprotein usually made of lipid plus protein and the lipid portion constitutes the triacylglycerol, cholesterol ester, free cholesterol and phospholipid and lipid portion usually apoprotein A apoprotein B48, apoprotein B100, apoprotein C and apoprotein E. So, classification of lipoprotein, there are 4 types of lipoprotein. These are mainly 4 types of lipoprotein. These are chylomicron, very low density lipoprotein, low density lipoprotein and high density lipoprotein. As we as in the slide, you are seeing that the size of the chylomicron is very big. That means the chylomicron is the biggest one, but density is the lowest, and high density lipoprotein is the lowest sizes lipoprotein, but density is more. Function of lipoprotein serves to transport lipid. As I have said before, this lipoprotein usually used to transfer the lipid from one organ to another. So, function is the serve to transport lipids in blood from one tissue to another, and importances, it is the clinical importance, how we use the lipoprotein clinically or in the investigations. Like estimation of serum VLDL, LDL and HDL has diagnostic importances in coronary heart disease, hypertension and atherosclerosis. Very common test known as serum lipid profile, we always give the patient to know the lipid status in our body. So, what we see in the serum lipid profile, that is the serum cholesterol, serum total cholesterol, serum HDL, LDL, this is usually seen in the lipid profile test. And fatty acid. So, this is all for today. Thank you so much for your patient sharing.